Yeah, so welcome everyone. Thanks for joining despite the sunshine out there. So it's really my great pleasure to welcome Sarah Teichmann, who's joining us today for this GHD lecture series. It's really great that we also not only talk about infrastructures and how to move data, but actually what to do with the data. So it's really great that we have Sarah here. Just a few words. Sarah did her PhD at the MRC, Laboratory for Molecular Biology in Cambridge, where she also started a group then later, before she moved to the EBI in 2013. That's where we met, a neighboring group. So really great pleasure to have you here. Sarah then moved on to the Wellcome Sanger Institute, where she started a program on cellular genetics that was very successful and also led much of the foundation of the Human Cell Atlas, a great initiative. And I'm sure we'll hear more about that today. And she very recently moved to Cambridge University to the Stem Cell Institute, where she's professor in gene expression genomics. So Sarah, thanks very much for joining us, and we look very much forward to your talk.
Thank you, Ollie, for the kind introduction. And if you want me to share, then you need to stop sharing, I think, and then I'm able to start sharing.
Thank you, Anna. If you can.
Yeah. Great.
Yeah, now it works.
Yeah. So as Ollie said, yeah, we were sort of office neighbors, kind of in the era of the beginning of the single cell era, I'll say, and then the early kind of starting up of the Human Cell Atlas in sort of 2012 to 2016 or so. So what I'll talk about today is a focus on really the analysis technologies for the Human Cell Atlas, after giving a short introduction to the initiative itself. And you know, just as for context, basically, the molecular definition of the cells in our body is really also answering the question of how a single genome sequence, since the DNA is nearly sort of identical in every single cell in our body, how does that single genetic code encode for this multitude of cell types? And of course, what we know now is that there are sort of on the order of magnitude of 10,000 or so cell types in the body, but there's a single genetic code. And so we're really asking how those programs are generated in our tissues. And this idea of making molecular maps of the cells in our body has a long history. And in fact, in Sidney Brenner's Nobel lecture over 2 decades ago, he said we need a program of making maps of cells and maps of how cells talk to each other. The cell map project, for which we don't need a model organism. So what he means by that is that we can study ourselves, our own tissues and organs will be one of the things to occupy us for the next few decades. And at that point, you know, the question was clear, but the way to get to the answer wasn't really clear. But by 2016, when we launched the Human Cell Atlas as an international consortium to create this reference map, the technologies and the way of getting to that answer was becoming much clearer. And the reason for that was that, of course, we had the resolution revolution in genomics and the technology developments that allow us to sequence the whole transcriptome, epigenome, to some extent also genome of individual cells and nuclei. And of course, that sort of developed a pace. And that technology, along with spatial genomics, you know, which allows us to identify and map the transcriptome in 2D and in consecutive sections in 3D, that sort of suite of technologies, along with the data science, the amazing AML techniques, the deep learning, you know, that I got exposed to as Ollie's neighbor in EBI, you know, that are so incredibly powerful. Those are really the technologies that are buoying the Human Cell Atlas initiative and making all this discoveries possible. And the consortium is an open kind of bottoms-up grassroots scientist-led initiative that everybody is welcome to join. You can sign up at humancellatlas.org/join-HCA. We have ongoing meetings, including one in Milan in September, if anybody is interested in coming to Italy at the end of September and see the new Human Technopole Institute with Piero Cornenci and Nicolo Soranzo and others. You're welcome to come to the meeting. There are still places. If you look at the Human Cell Atlas website, and you can see that now we have over 3,000 members from almost 100 countries around the world. We're organized into working groups in terms of the technologies and analysis and ethics and equity, and also biological networks. So the question kind of that we some of the questions that we can address with the Human Cell Atlas is not only what are the cells in our body, what are the expression programs that are encoded by our genome, but also how can we use that for informing, for instance, programming of cells in vitro. And one example of that is the tracking the pathway of T cell development from hematopoietic stem cells in the thymus, and the Thymus Cell Atlas, which we've worked on in collaboration with other groups, and sort of published in 3 papers defining the cells in our developing and postnatal and adult thymus. And of course, we can use that data for engineering cells, both from the point of view of the cell intrinsic program and the transcription factors that are needed for making the T cells and generating them from iPS cells, but also in terms of the tissue niches and the signals that the cells are seeing as they're traveling throughout the different niches in the thymus. What is the extrinsic signaling, and can we learn from the in vivo thymus to program the cells in vitro by adding the same sorts of factors in the dish? And so in that sense, the Human Cell Atlas is like a blueprint for engineering cells in the dish, and that's kind of the funny picture kind of showing that. I mean, how can we quantify that using data science methods? Well, one tool that's really useful for this is trajectory alignment, and basically it's quantifying the similarity in cell states in the in vivo trajectory, for instance, versus the in vitro trajectory. Of course, this could also be healthy versus disease trajectory, or rodent versus primate cell trajectories, and so on. So having the quantitative gene level alignment, so not only the cell level alignment in terms of matching the cells to each other in different cell states, but also having the gene level alignment of single cell trajectories is really useful in telling us which transcription factors we have to add, which signals we have to add into the dish when. And a Bayesian sort of information theoretic framework to do this at the gene level for the first time instead of at the cell level is something that we released recently on bioRxiv. It's a collaboration with Bianca Dumitrescu and others, and it was driven by a really talented computer scientist in the group who's a Schmidt Accelerate Fellow, Dinithi Sumanawera. The Python package is on GitHub, and this is part of the Human Cells publication bundle that will come out sort of in the autumn, and is in press at Nature Methods. So I encourage you to kind of take a look at that if it could be useful to you for any trajectory comparison in terms of linear cellular trajectories. We're now sort of thinking about developing this further into a more general framework. Another kind of key data challenge that we're facing in the Human Cell Atlas initiative at the moment is the challenge of data integration in order to get towards a reference data set, at least for suspension cell data. So, what I mean by that is that for any given tissue or in any given organ, let's say in the airways, the gastrointestinal tract, the thymus that I showed you, heart, and so on. Many groups are working on these tissues and generating data, and have done over the past decade or so. And so what will be really useful for the Human Cell Atlas community, but also the general biomedical community, will be to have a sort of assembled gold standard reference with cell annotations and integrated data sets. And of course, there are many ways of tackling this in terms of annotating new data sets. What we provide for the community is a collection of logistic regression models that can rapidly screen kind of against a new data set, provide a fast first pass annotation. And the way this works is that we have a website at celltypist.org. You can either upload data or download models, and then there are models for all kinds of collections, for tissue-resident immune cells, for airways, gastrointestinal tract, cardiac, and so on and so forth in this database. There's also a searching function for basically searching for expression of a given gene across different data sets, or expression or searching for a cell and where it's expressed in the human body. And to make these logistical regression models, what we need is integrated kind of correspondence between different data sets. And the way Chuan Xu and the group did this was to cross-match against, relate different data sets to each other with logistic regression, and then allow us to basically harmonize the annotation so that we can then model each cell type kind of as a with a given word or label attached to it, if that makes sense. And so these two tools are available online on celltypist.org and on GitHub. And Chuan used the CellHint framework that he developed recently for also analysis of different neural progenitor populations in the human brain, including sort of neuroblast cell states in different regions of the brain, using the brain cell atlas data that's of course published recently in Science as a fantastic publication bundle and as a resource for the community. And so this integration kind of challenge or exercise is something that that many groups in the community are working together on at the moment, and there's kind of coordination of this by Malte Lücken in Helmholtz Munich, and the sort of common pathway or roadmap that each project is following is basically defining the scope and the data sets involved, processing metadata. And so this is pretty important to know is, for each sample, which lab does it come from? Is it from a different lab or from the same experiment? Is it from is there metadata about the donor or a patient or donor sample, QCing the data, cell label harmonization, which is what CellHint does, in an automated or semi-automated sense, what I just mentioned. Then integrating different kind of different automated, different frameworks or different methods with different parameters, so that you land on the most optimal framework, and then proceeding basically to the full data integration, clustering, marker detection, rechecking the annotations together with the biological experts, the human cell as bionetwork, and then that gives you the framework for downstream analysis. And the sort of the first poster child for this was the Human Lung Cell Atlas. And here we had, just to give you an idea, 500,000 cells from 14 different data sets, 107 individuals. Many different protocols were used for generating the single cell data. There were 166 different samples, and then there are different anatomical locations from the upper airways to the lower airways, like including nose, trachea, bronchi, bronchioles, parenchyma, and so on of the lung. And that sort of gave us the core, which was then extended by transfer learning to include disease data sets. And those then had almost 2.5 million cells from almost 500 individuals. You can see in terms of metadata, the age, sex, smoking, BMI, and so on, are kind of interesting to sort of correlate with different features of the cell composition, essentially in the sample, as one example. So there are many different applications, basically, of this kind of integrated reference object in terms of then annotating new data sets, discovering disease-associated states by using the healthy reference as a framework, and so on. And so this is what, as a community, we're pursuing now for 18 different biological networks, so that we have for all of those basically a reference. And on data.humancellatlas.org, you can see the brain basically released, which is based on that Brain Cell Atlas publication bundle from the NIH Brain Initiative and Human Cell Atlas collaborators, and the lung, which I'm showing here. And this was driven by Lisa Sikema and Fabian Theis and the lung bionetwork community. And then there's an eye. I think there's an eye object for the retina that's being released. And then there's many others that are in the pipeline: the blood, the breast, pancreas, liver, kidney, heart, et cetera, et cetera. So these are all going to be kind of Human Cell Atlas collaborative efforts to provide these gold standard suspension cell and nuclear data sets for the community with harmonized consensus cell annotations. So as in that, for that purpose, one of the challenges that we have, or a future kind of challenge that's on the horizon, is, of course, that we want to take that suspension cell data and then integrate it with other layers like epigenetic, sort of open chromatin layer, spatial data that I've showed, large data sets with human diversity. And one framework that we've developed for tackling kind of the multi-omic modeling is this deep generative decoder framework that we call multi-DGD, which builds on a deep generative decoder for RNA from Anders Krog's group. And this was developed by Victoria Schuster, who came to visit our group from Copenhagen, from Anders' lab, together with Emma Dam, who's a joint student between my lab and John Marioni's lab. So the benefit of the deep generative decoder is that you have this shared representation for the multiple layers, and you're modeling basically the specific modalities in lower layers with a branched architecture, as well as modeling covariates, which I'll come on to. So the performance of this deep generative decoder framework is that it can be trained on fewer cells with the same sort of quality of integration. And here's a benchmark against another model that does a similar thing, multi-BI from Mir Youssef's group. And this is basically a bone marrow data set with 13,000 genes expressed and 116,000 attack peaks from 70,000 cells with about 22 kind of coarse grain cell types. So the performance, of our method stays closer, basically so this is trained on the full data set, and you can see here that the benefit of multi-DGD is that it keeps that performance even on smaller fractions of data. So you can basically train it kind of more sensitively and faster without loss of performance. So the benefit of DGD is that not only that it models these multiple modalities, but also that we can model, that we can improve kind of the deep generative decoder itself by modeling multiple covariates and disentangling them. And so multi-covariates means things like age, disease status, for instance, could be covariates. Batches could be covariates. So you can basically model them explicitly and then separate that type of information in a process called disentanglement. So the covariate model is something that's new in this application, but we also wanted to lay the foundation for using what's learned by the model to learn about how modalities are connected. And so in this case, we are linking chromatin, accessible chromatin regions with distal enhancers, distal repressors, et cetera, that are shown here in yellow and orange to the expression profile of a gene. So we're looking into whether the changes in a specific gene count are linked to changes in open chromatin and other regions of the genome. And basically, the question that we're asking is here, can multi-DGD predict promoter-associated genomic regions through backpropagation through the model, through the deep generative decoder model, by in silico kind of simulating perturbations. So we here what we're doing is taking all of the data as input and training on it, and then basically in an in silico experiment asking how the sample will change if a gene is switched on or off. So we're making that single small step, and then looking at the prediction in this generative model. What would the generative model predict if you have this perturbation? And what we've done so far is looking at basically whether, so one simple thing that we tested was whether you get changes when you silence genes. Do you get changes close to the promoter or far away? And what you can see here is a kind of common sense test, is that most of the changes the model predicts are basically close to the promoter. So that makes sense in the sense of promoter regions and proximal enhancers changing. The other thing that we checked was whether we can see any link between distal regions and genes. And of course, that's kind of a much bigger challenge. And a more open question in biology is, how do distal regulatory regions kind of control promoters that are, that are not right around the transcription start site. And that's, can be interrogated experimentally with things like high C or high chip. And so the reference data that we use here is, I think it's from ENCODE. It's a data set on T helper cells, so CD4 T cells with high chip, that tells us about the connection between promoters and distal enhancers in the experimental CD4 T cell data. And so what we did was basically train on the, I think it was the bone marrow data, or the, so train on a basically an RNA attack, I think CD4, yeah, it was CD4 T cell multiome data, and then see whether with our perturbation model we can recapitulate the links that are observed experimentally in the CD4 T cell data. And we have a negative control here with high chip. And basically the conclusion from this was that we can see stronger effects in regions that had high signal in the experimental data. So we do find what we're looking for, like in these gray regions, we do see the perturbations that we're looking for. But we do also have other associations that weren't found in the experimental data that we don't know sort of what they're saying. You can see in the red here, for instance. So this is kind of another pretty cool feature of generative AI, is that it could allow us to predict these higher order relationships between promoters and enhancers, that are sort of implicitly encoded in the multiome data, but not measured kind of directly. And so you can, but it's a very exciting kind of potential in that sense. And this is the preprint, and the GitHub link was there before, and we encourage you to play with this data, with this kind of software, and this will also hopefully be Nature Communications in the Human Cell Atlas paper bundle that will be coming up. And so that really brings me to the end of the talk. What I summarized was kind of the use of genes to genes as a gene level alignment framework. This is a Bayesian framework for dynamic programming alignment of gene expression patterns, cell typist and cell hint for automated cell annotation and kind of label harmonization, and then multi-DGD as a generative AI framework for modeling and predicting relationships in RNA and attack data. And I will stop there and say thank you and take any questions.